Imagine a microscopic battle happening right inside your body, a fight where the stakes are high, and the soldiers are highly specialized proteins known as antibodies. These warriors are at the forefront of your immune defense, designed to seek out and neutralize invaders like viruses, bacteria, and toxins. But what exactly do antibodies look like, and how do they carry out their mission to protect you? In this journey, we'll take you deep into the world of antibodies, revealing their structure, function, and how they play a critical role in your body's defense system. At a basic level, antibodies are Y-shaped molecules, resembling a Y with two arms extending outward. Each arm of the Y is a highly adaptable structure capable of recognizing and binding to a specific part of a pathogen, called an epitope. An epitope is a unique molecular signature found on the surface of an invading pathogen, whether it's a viral spike protein, a bacterial cell wall component, or a toxin. This binding process is crucial, as it marks the pathogen for destruction and prevents it from causing harm to your body. Imagine the arms of the antibody as being like a pair of perfectly shaped keys that fit only one specific lock, and that lock is the pathogen's unique epitope. The Y-shaped structure of the antibody is made up of two heavy chains and two light chains. These chains are held together by disulfide bonds, ensuring the antibody remains intact. The variable region of the antibody, which forms the arms of the Y, is the part responsible for recognizing the pathogen. This region is highly diverse. Each antibody can have a unique variable region, allowing the immune system to create a vast array of antibodies, each tailored to target different pathogens. The more diverse the antibodies, the more equipped your body is to fight a wide variety of infections. The other part of the antibody is called the constant region, which forms the base of the Y. This region doesn't change and is the same for all antibodies of the same class. The constant region is what allows the immune system to differentiate between the different classes of antibodies, IgG, IgA, IgM, IgA, and IgD. Each class plays a specific role in your immune response. For example, IgG is the most common and versatile antibody in the blood, circulating throughout the body and providing long-term immunity. IgA is found in mucosal areas like the lungs, gut, and eyes, protecting these entry points from pathogens. IgM is the first antibody produced in response to an infection, acting as a quick and powerful initial defense. IgA, often associated with allergies, is involved in defense against parasitic infections, while IgD is mainly found on the surface of B cells, where it plays a role in initiating immune responses. The beauty of antibodies is their specificity. Each one is created by B cells, a type of white blood cell. When a pathogen enters the body, B cells encounter the invader and, with the help of other immune cells, start producing antibodies tailored to that specific pathogen. This process is known as clonal selection. A single B cell can recognize one pathogen and, upon activation, rapidly divide to produce a clone of cells, all secreting the same type of antibody. Over time, the immune system fine-tunes its response, and memory B cells are created. These memory cells remember the pathogen, allowing your body to respond faster and more effectively if the same invader strikes again. This is the basis of immunological memory and is why you typically don't get sick from the same virus more than once. So, how do these antibodies fight? Their main role is to neutralize pathogens and flag them for destruction. Once an antibody binds to a pathogen, it can prevent it from entering your cells by blocking the surface proteins that viruses or bacteria use to latch onto host cells. For instance, in the case of viruses like influenza or coronavirus, antibodies can bind to the viral spike proteins, effectively preventing the virus from docking with a host cell and initiating infection. Antibodies also act as recruiters. Once they bind to a pathogen, they can attract other immune cells, such as phagocytes, like macrophages, and natural killer, NK, cells, to come and destroy the pathogen. This is called opsonization. The antibody essentially coats the pathogen, making it easier for other immune cells to recognize it and eliminate it. This is where the complement system comes into play. Once an antibody binds to a pathogen, it can activate a group of proteins known as complement proteins. These proteins help destroy the pathogen directly, either by puncturing its membrane or by marking it for immune cell attack. Antibodies don't just neutralize viruses and bacteria, they also help to prevent the spread of infection. By binding to viral particles or bacteria in the bloodstream or tissues, they can prevent these invaders from reaching new cells. This is why antibodies are so important in protecting you from getting seriously ill or from spreading the infection to others. But viruses are clever, they don't just sit back and let the immune system win. Many viruses have evolved strategies to escape detection by antibodies. For instance, the HIV virus mutates its surface proteins rapidly, making it difficult for antibodies to keep up. Influenza also undergoes frequent changes in its surface proteins through a process known as antigenic drift, which allows it to evade recognition by the immune system, making it necessary to update flu vaccines every year. In response, the immune system evolves as well, producing more antibodies and increasing their affinity for the virus over time. Interestingly, antibodies themselves are being used in medicine. Monoclonal antibodies are laboratory-made antibodies designed to target specific pathogens or disease-causing molecules. For instance, during the COVID-19 pandemic, monoclonal antibodies were used to help treat infected patients by blocking the virus from entering cells, effectively reducing the severity of the illness. These engineered antibodies are becoming a key component in the treatment of many diseases, offering new hope for treatments where conventional therapies might fall short. Antibodies are also crucial in vaccination. When you get vaccinated, your body is introduced to harmless pieces of a pathogen, such as inactivated virus particles or viral proteins. This teaches your immune system to recognize the virus and produce antibodies against it. If you're later exposed to the real virus, your immune system is already prepared to fight it off, using the antibodies it created during the vaccination process. This is how vaccines protect you without causing you to get sick from the disease. In essence, antibodies are the body's specialized soldiers, designed to fight off a never-ending array of potential invaders. Their unique structure, ability to recognize specific threats, and capacity to recruit the rest of the immune system make them one of the most powerful weapons in the body's defense arsenal. Whether neutralizing a virus, marking a bacterium for destruction, or even preventing future infections, antibodies play a critical role in keeping us healthy. And in our fight against emerging diseases, their ability to evolve and adapt is what gives us the upper hand in an ever-changing battle.